Good afternoon to all. Today we are going to discuss about structure of bacterial cell wall. I am Dr. H. Vajiha Banu, Assistant Professor, Department of Microbiology, Jamal Mohammed College, Trichy. Cell wall. Cell wall is the outermost layer of cell. In many cases, the cell wall comes in direct contact with the environment. Then the functions of cell wall: it protects the cell, it maintains the shape of cell, then it maintains the osmotic integrity of the cell and assists some cells in attaches to other cells. It provides attachment site for bacteriophages. It plays an important role in cell division. It provides a rigid platform for surface appendages like flagella, fimbriae and pili. Next, the peptidoglycan. Peptidoglycan, also known as murin, is a polymer consists of sugars and amino acids that form a mesh-like layer outside the cell membrane of most bacteria, forming the cell wall. The sugar component consists of alternating of beta-1,4 linked N-acetyl glucosamine and N-acetyl muramic acids. These subunits, which are related to glucose in their structure, are covalently joined to one another to form a glycan chains. They are attached to the N-acetyl muramic acid is a peptide chain of 3 to 5 amino acids. The peptide chain can be cross-linked to the peptide chain of another strand, forming the peptidoglycan. This is the structure of peptidoglycan. Next, gram-positive cell wall. Gram-positive cell wall usually thick in nature, homogeneous, composed mainly of peptidoglycan. It accounts for 50 to 90% of dry weight of the cell wall. It contains large amount of teichoic acid. This is a structure of gram-positive cell envelope. Then these are the components of gram-positive cell wall. First one, teichoic acid. Teichoic acids are connected to either peptidoglycan or to plasma membrane lipids. Teichoic acids were absent in gram-negative bacteria. Then functions of teichoic acid: it acts as a receptor molecule for bacteriophages. It participates in the supply of Mg to the cell by binding magnesium ions. It regulates the normal cell division. For most part, protein is not found as a constituent of gram-positive cell wall except M protein on group Streptococci. Then gram-negative cell wall. Gram-negative cell wall is multi-layer and more complex than gram-positive cell wall. Peptidoglycan of gram-negative bacteria is thin and contains 10% or less of cell wall. The outer membrane lies outside the thin peptidoglycan layer. So most abundant protein present in gram-negative cell wall is Brown's lipoprotein. This is a structure of gram-negative cell envelope. Then special components of gram-negative cell wall. First one, periplasm. That means the region between the cytoplasmic membrane and the outer membrane is filled with a gel-like fluid called periplasm. In gram-negative bacteria, all secreted proteins are contained within the periplasm unless they are specifically translocated across the outer membrane. Periplasm is filled with the proteins that are involved in various cellular activities including nutrient degradation and transport. Next one, outer membrane. Peptidoglycan layer is surrounded by outer membrane in the gram-negative bacteria. It's made up of lipopolysaccharides rather than phospholipids. For this reason, the outer membrane is called the lipopolysaccharide layer or LPS. The outer membrane functions as a protective barrier and exclude many toxic compounds. Lipopolysaccharide molecule is extremely important from a medical standpoint. It consists of three parts. Two of them are medically significant: lipid A, core polysaccharide, then O antigens. Lipid A embedded in membrane. Core polysaccharide is located on the surface of membrane. Then O antigen, which are short polysaccharides, extend out from core. Lipid A. The chemical makeup of lipid A molecules play a significant role in our body ability to recognize the presence of invading bacteria. It is toxic in nature. As a result, the LPS can act as an endotoxin, causing symptoms like fever, diarrhea and shock. Then O antigen. O antigen, it is composed of carbohydrates including glucose, galactose, mannose and some other sugars in varying combinations. The O antigens can react with their specific antibodies. These are the structures of bacterial cells. Thank you.